music of the spheres is music to my ears. It's soft melody, it's soothing, moving me to tears. I woke up in a cave, I saw the shadows on the wall and the shackles on the saw.
Actually, I'm on the board of directors for Beltway Atheists, and it's a nonprofit here in a Loudoun area, and we do a lot of different things. Just to put in a little plugging for that. Part of what I do is I try to reach out to high school students and try to foster or help them foster an environment where they can start like atheist groups. So I do a lot of that with Beltway Atheists, and I also reach out to college campuses. I started an atheist organization at VCU that about 6-7 years ago, and it's still going strong, so that's good. I'm helping those guys out, kind of get speakers and things like that. So, and then every so often I do these little talks. I know Nicola here, I've given this similar talk to this at her school at Dominion, and she is helping out a free thinkers group down at Dominion, so I've come there and do stuff with her. So anyways, we do have a social wing, Nova Atheist, which is on Meetup, and we do mostly social activities for that. So if you guys are interested, check this out. So a little bit of background for me. I was actually born and raised in Italy as a Catholic, and as most Italians do. But about age of 20, I realized, I got really interested in biology, and I decided this religion thing is not quite working out for me. So I kind of left the faith and became active active in the atheist community about 7-8 years ago, seeing some of the things going on. So I decided to become part of the community. So that's my background. I have a master's in biology. I studied malaria parasites, which is a topic that we're going to talk a little bit about today, and somewhat a little bit of detail, which is kind of a neat story with that. So the talk is evolutionary creationism. What controversy? And that's actually an interesting question, because is there really a controversy? I guess it depends on how you define controversy, right? In the scientific community, there is no controversy. In the social and public arena, there's a big controversy. And we'll talk about a little bit of the reasons why. So I'm going to give just some broad general topics that I'm going to talk about is this idea of science versus God, and what that really means, and does it really even mean anything. Defining science is a big thing, because I think right now defining science and what theories are is what is causing this controversy in the public arena. I mean, how many times do we hear, "Oh, it's just a theory"? Okay, well we're gonna go ahead and define what this theory actually really means. We can't let the other side define what the theory is. And then at the end, the second half of the talk, we'll talk about creationism and some of the creation science things going on right now. Maybe compare some of these things with evolution, and I'll spend a lot more time with the intelligent design, because that was the latest and newest wing of creation science, the intelligent design. So all right, so God versus science. This is how the media is portraying this issue. Okay, evolution wars. In my opinion, is that this idea of God versus science was really started by the Christian right, because science was never really developed to actually destroy religion or God or anything. It's just science. It is what it is, and it's really only when it comes to evolution that this becomes a big problem. And we have a lot of theories out there that nobody seems to really have an issue with. Nobody has an issue with the wave properties theory of light, or nobody seems to have a problem with gravitational theory. Why is evolutionary theory such a problem? And why is it now being labeled as God versus science? Well, I like this quote from Simon Conway Morris. He's a paleontologist at the University of Cambridge. He says this: "Too much of modern-day evolutionary discourse is dogged by streniency that in other areas of science would be regarded as astonishing," right? Nowhere else do we see this kind of anger in science. To some extent, this is a reflection of the growing influence of so-called intelligent design. And that is true. And because intelligent design is a crafty little way to really trick people into thinking it's actually real science and actually a good opposing theory to evolution. But the end goal of intelligent design is actually religious in nature. It really is. I mean, there's no doubt about that. And what this does is it combines the worst of all possible worlds, being non-science, this intelligent design, but also flawed theology, which is really important. Because if you're a creationist and you believe in the biblical story, intelligent design is actually flawed theology. I mean, you got to evolve your theology to kind of fit modern science. And here's the interesting thing about this. There are Christian apologists that actually, that's what they do, is they try to rework and evolve religion to fit what modern science finds out. I guess you guys have heard of, God of the Gaps, and a new one that I think it was either, I can't remember who coined it, I think it was John Shook or Bosch, I think, but coined theology at the edge. In other words, where God is kind of hiding now at the edges of science, of the unknown. But yeah, and this is, in regards to things like evolution and also cosmology to some extent. But here's the real reason why evolution is under attack. And this graphic was taken from Answers in Genesis. Is everybody familiar with Answers in Genesis? It's Ken Ham. He's the guy that, he's a young earth creationist who built that creation museum and is also in the process of building that Noah's Ark Adventures, like an amusement park based off of Noah's flood. And he actually got some tax credit for doing this, okay, which is unbelievable. But anyways, what these people think is this. God's Word, the foundation of God's Word, is creation. That's at the beginning of the Bible. And everything else, okay, goes up from that. You have from creation and God's Word, and you have laws. But with opposing that, diametrically opposed to this, and this is how they're setting this thing up, is you have man's opinion, right? Evolution is man's opinion. That's all it is. And from that stems lawlessness, clearly, because everybody who accepts evolution as a true theory obviously is going to be, a criminal and going around and doing things like, raping and murdering and killing. And then, of course, from these laws stems this idea of marriage, family and marriage. And of course, we have this evil homosexual behavior going on, right? Man's opinion, and this is what evolution leads to, okay? And then, of course, standards, and of course, pornography, because everybody who accepts evolution is stuck to a computer looking at porn all night. And of course, this idea of the meaning of what life means, and of course, that leads to, all abortion. And so this is kind of why, people say, well, if we really show these creationists, if we really explain it to them in a way they understand it, wouldn't they accept evolution? Well, no, they're never going to accept it because they think these evolutionary termites are crumbling their creation story, which then is going to crumble the rest of their ideals. So this is really what we're talking about. This is why we have such a problem, and it's going on today. And this is why evolution. And to show you another graphic here, this is again from Answers in Genesis. It's another graphic on this war that they've set up, this cultural war that it all really stems. These people really believe it stems from evolution and everything else. These what we hear in the media all the time. As they're trying to pop these balloons of abortion and gay marriage and all this. We're here chipping at their foundational beliefs. I like here how they put that humanism flag. So because apparently humanism is what's leading the charge here of all this behavior, right? So, but that's how they're viewing it, and that's the problem. This is why evolution's under attack. So the First Amendment prohibits public schools. So, one thing, the First Amendment prohibits public schools from teaching religion. Okay, that was established. Okay, that's clearly, it's the Establishment Clause and things like that. But this is why creationists now are trying to insert creationism in school by disguising it as science. So this is not why we're seeing these things like intelligent design and creation science. Question is, does this pass muster? And we've seen, we know about the Dover, the Pennsylvania trial, and that the intelligent design died at that court decision. So my friend W here, asks the question. Really, is the question asked: is our children learning? Is our children learning? That's a very important question because that's, our public schools, it has a lot to do with what our children learn. So I want to go ahead, and what I'm going to do now is kind of switch gears and talk a little bit about what science actually is, all right? And then go on there, kind of try to give some examples of, and then get into like theory and some of the examples of evolution that we're seeing today. So, first of all, what is science? I mean, what really is science? Well, it's a lot of things. We have all kinds of different fields in science: geology, biology, chemistry, physics, and all these. And what all these fields are trying to do is they're trying to understand our physical world. So science, if you look at the dictionary, because when you define something, the dictionary is the best place to look, right? Science comes from scientia, which is Latin for knowledge. That's all science is knowledge. It is knowledge that's covering general truths of the operation of general laws, especially those obtained and tested through the scientific method and concerned with the physical world. So these are the key words here. Tested through scientific method, and concerned with the physical world. Okay. Science does not concern itself with the supernatural because it doesn't have to. Okay, and it's not really designed to do that. So let's talk a little bit about the scientific method and what it actually is. The steps of the scientific method are as follows. You have observation, so you notice something in nature. When I did my graduate dissertation, I looked at a malaria parasite and one of the proteins that it secretes. And that was an observation. What does this protein do? They sequenced, actually, it came from a sequencing of the malaria genome, which was a huge project and is now helping out tremendously as far as malaria research. But so you find this protein there. Okay, what does this do? And that was my observation. And when I was reading upon it, I found out that this protein is part of a family of other proteins that do a number of different things, but one of them is recognize red blood cell surface receptors, and they bind to it. So that was my observation. I did some literature search, and I said, okay, I'm going to develop a hypothesis now. Now, hypotheses are different than theories. Okay, hypothesis is a good estimated guess of what you think something is going to do or what this observation is. Whereas a theory, as we're going to find out, is something a little different. So I'll make a hypothesis and say, okay, this protein is going to probably bind to some kind of cell surface receptor because it's part of that family of proteins. So okay, so this is my experimentation and collected a lot of data and analyzed that data. And then I arrived at a conclusion. I said, you know what, aha, I did. I found it. This does actually bind to a cell surface receptor. And so that is going to be my conclusion. And to give you the short end of it, that is what it is. So that's how it works. All right? But more important, this method works well. And if you set this method up right, you actually set it up to kind of prove yourself wrong. Because if you fail to prove yourself wrong, then that means the only other choice is that you're right. That's the idea of a null hypothesis, trying to prove or disprove a null hypothesis. But more important than that is we have this thing called predictive value. Because based off of the scientific method, and then once one of the big things about the scientific method, if you follow it properly, it has to be reproducible. So if it's true, that means a million other people that do this have to come to the same conclusion. Otherwise, your research doesn't work. And in science, this happens all the time. People set up an experiment, they publish a paper, and then somebody reads the paper and goes, let me try to repeat this in my lab, and it doesn't work. And then they write a paper going, this guy either messed up or falsified data. So people get caught a lot in science about this. But this idea of predictions is actually very important because if you can make predictions, and those predictions are right, you know you're doing something right here. And we see this, and I'll give some specific examples here. One of these, this idea of predictability is the idea, can evolutionary theory predict, something called convergence evolution, okay, or convergence. What convergent evolution states is that given a similar environment, for example, in the case here, like marine vertebrates, given the similar selective pressures, they are going to actually evolve to sort of look very similar. So you have here a fish, an extinct, reptile, ichthyosaur, and a mammal. And if you look at their body shape, these all arrived independently. These aren't part of the same evolutionary lineage. But somehow evolution was able to, natural selection was able to kind of shape these vertebrates to look pretty much similar. And that has to do with the idea of convergent evolution, meaning converging into phenotypes and physical body structures all similarly. And the key thing behind convergent evolution has to be independent, right? So what was valid millions of years ago is still valid today. Another example of convergent evolution, we can, how this ties into predictability is we can actually, kind of guess and tell how these animals are going to look. And another example here is like these are placental mammals here, right? We're all placental mammals. You have like things like flying squirrels and groundhogs and anteaters, and this is a mole and a mouse. We have in Australia marsupials that, if you know anything about Australia, kind of split off from the, remember if it was Pangea or some of the other ones, but it split off, and all the animals that were stuck in Australia and some of the islands surrounding it evolved in a completely different manner. They went a completely different route. Their reproductive strategies have all changed. So these guys are marsupials that they're completely different types of mammals. However, they still evolved to look similar to some of the animals that we have here. So this is a flying squirrel. Flying squirrels they live out in trees and they jump from tree to tree. They developed a mechanism where they can actually flap out their skin and they can glide long distances still from tree to tree. Clearly this has a lot of advantages when they're looking for food. We have marsupials that do the exact same thing. They almost look identical. Okay? Like sugar gliders. You guys have seen sugar gliders? I just recently got a couple of them. They're kind of neat little marsupials from Australia. But that's exactly, they look almost identical to our flying squirrels. Same thing, we got our groundhogs and the wombats. Short, stocky bodies. They live in similar conditions. Anteaters. So these marsupial anteaters exploit the same type of feeding behavior as the anteaters elsewhere. And of course you have our placental moles and you have marsupial moles that look exactly, they evolved similar characteristics. And this all has to do with something called toolkit genes, which we'll talk about here in a minute. But again, we can predict what animals are going to look like, how they're going to behave, how they're going to eat, based off of this idea of convergent evolution. So this makes sense, to some people anyway. So anyways, another aspect of predictability. You got two kinds of whales, right? We got baleen whales, and you got toothed whales. Well, somebody predicted, obviously, if you follow evolutionary theory, that they had to have had a common ancestor, a transitional fossil, so to speak. Of course, I'm told repeatedly that there's no such thing as transitional fossils, which I have no idea where these people are getting this from, because we have hundreds of thousands of transitional fossils. But somebody predicted that there had to have been a transitional fossil that showed characteristics of both teeth and baleens. Well, this was found in 1966, and it's called Etecedus. And it lived about 25 million years ago, and that's kind of what it looked like. So this was a transitional fossil as these two lineages were splitting, the baleens versus the vertebrates. Again, predictability. So I'm going to switch gears and talk a little bit about something called empirical falsifiability. Has anybody heard this term before? Okay. It was developed by Karl Popper, who was a philosopher of science. And he makes a claim, he postulates that a theory should be considered scientific if and only if it is falsifiable. What falsifiability means is that it has to have a logic possibility that an assertion should be shown false by a particular observation or physical experiment. In other words, in order to call something a scientific theory, or call something scientific in general, there has to be a possibility that it's not that you can prove that it's not true, like without a doubt. So the question then becomes: can creationism be falsified? It's a good question. What about God? Can God be falsified? Probably not, because there's no way to absolutely prove, at least right now, that God does not exist. I mean, we got to be honest. There's absolutely, there's no reason to believe. The same way there's no reason to believe that Santa Claus exists. So there's really no reason we can't make an absolute statement about them, right? But creationism, on the other hand, I don't know. But I don't know, if you guys are familiar with Victor Stenger, I know some of you guys know Victor Stenger, right? Well, Victor Stenger actually does say, he actually does make that statement that we have actually empirically proven that God does not exist. I'm not quite sure I agree with that, but with creationism, I'll leave that question open and we'll see. We'll come back to this here in a bit. So, yeah, of course, you can't prove God don't exist, right? So, eat it, Darwin. And, it's kind of like, that's kind of how you got to deal sometimes. So, let's talk about scientific theories and what they are. A scientific theory is a set of facts. Okay, that's the key words here, set of facts in their relation to one another. Okay? How these facts kind of come together to form a broader picture. That's what this scientific theory actually really means. It's not something that somebody just, hey, I got an idea. Darwin didn't sit there and go, hey, evolution, let me go out and look for evidence towards it. He had no idea what that was when he first started his journeys and his studies. He actually was more interested in geology at the time than anything else. But as he then developed, he started studying things. He just couldn't help but put these things together, these sets of facts, which then allowed him to make this theory. And it's also defined as a plausible or scientifically acceptable general principle, body of principles offered to explain phenomena. So that's the big key word here. So what are some of these scientific theories? Well, we got a bunch of them. Big Bang theory, wave theory of light, and there's also the particle theory of light, right? Theory of gravitation. Gravity is just a theory. I get a lot of people, I do a lot of online debating sometimes, and when I'm really feeling kind of irky, I tell creationists and say, if you don't believe in gravity, why don't you go to the tallest building you find and take a leap of faith? And tell me if gravity is just a theory at that point. Because you can't see gravity, right? You can't see it. So why aren't these people out there trying to introduce intelligent falling? Because it's the same exact kind of idea, right? Intelligent falling. Hey, let's teach the controversy. I want to introduce intelligent falling in physics class and see how well that goes. Then of course germ theory of disease, right? If you really are creationists, you shouldn't believe in just a theory of germs, right? You can't see bacteria. Anyways. And there's always, relativity and quantum theories and things like that. And of course, then we come to evolutionary theory, which is the big thing. And what this is defined as, a theory that the various types of animals and plants have their origins in other pre-existing types, and that these distinguishable differences are due to modifications in successive generations. So it's descent with modification. Okay? And we'll get really now deep into these. I'll give you some examples here. So of course we can't talk about evolution and evolutionary theory without talking about Charles Darwin and natural selection. Now, Charles Darwin did not develop the theory of evolution, by the way. Okay, that was being discussed even in the days of Aristotle and Plato, different aspects of evolution. There was this idea already floating around, this idea of evolution. What Charles Darwin gave us was the mechanism of biological evolution. He gave us natural selection. That's what he did. And I'm not going to read this whole thing, but if you see some key words here, variations, okay, thousands of generations, advantage. And at the last part here, this preservation of favorable variations and the rejection of injurious variations I call natural selection. And that's what this is really all about. Right? So there's three main ingredients to natural selection, just three. Variation, which what this variation really means is genetic mutations. You and I have about 40 to 60 genetic mutations that your parents didn't have. And your kids have 40 to 60 genetic mutations that you don't have, and their kids are going to have, and so on and so forth. And that just happens when you're trying to copy billions and billions of DNA code. You're going to have mistakes, and that's where these variations kind of happen. These mutations in certain genes can then develop into certain phenotypes. Some are injurious, some are bad, some are neutral, and some are good, as we're going to see here in a minute. So these variations then are selected upon selection by perhaps predators. And then the third item is time. You need time for a population to actually evolve, right? So even though evolution is selected upon on individuals, it's the population. It takes time for it to evolve. So given this kind of example here, you have two color morphs of a beetle. You have a green morph and a sandy morph. And say this beetle lives in a desert, and so there's a lot of sand around. The birds are these guys are going to camouflage very well, and the birds are going to see the green ones, and they're going to pick them off and eat them. Well, eventually, as time progresses, you're going to have less and less green ones, more and more of these gold ones. These gold ones are going to reproduce, and then over time, this process can actually happen relatively quickly. And I'll give you here an example here in a minute how quickly this can happen. So these are three items you need. Just to kind of move into the human evolutionary history here. This is simplified stuff. This is not how we evolved. Okay, evolution is not linear. However, this kind of makes a point of, it's an old school thought, but really this is how evolutionary hypotheses are right now. So spanning back about 7 million years ago. The point I'm really trying to make with this slide is that we have different species evolve and co-evolve sometimes at the same time. When you look here where humans are, we actually share the Earth with other hominid species, other very intelligent hominid species like the Neanderthals. We actually share the Earth. And now recently they've published, there's a study that actually within at least some subsections of the human population, we have some Neanderthal gene because they believe there was some crossbreeding going on. So just to let you know that evolution is not linear. See, you have these things, and maybe there was a jump here, something happened. And so these guys kind of moved on, and then maybe they gave rise to some of these guys. And again, it's all about how these guys live and geographic differences and thinking how things evolve. So, but and actually, that's a correct statement. We don't come from monkeys. We don't. We really don't. Because if we did, why do monkeys still exist, right? But that's true. We come from progenitors of animals that kind of gave rise to both. We have the transitional fossil. We have a transitional form that led then to monkeys and the great apes and the hominid species. So that's actually a true statement. All right, let me switch gears here, and I'm going to give you now a couple of examples about how evolution actually works in real life, how it works in humans, and I'm going to call this section Malaria and Sickle Cells. Selection, predictability, and evolution, because this is actually kind of a very important aspect to show you kind of even the medical importance of evolutionary theory. So I'm going to start out with giving you a quick introduction to how malaria kind of works. You get bit by a mosquito. Mosquito injects the malaria, what they call sporozoites, which is the form of the malaria parasite. And by the way, malaria, the parasite is actually, it's not a bacteria, it's not a virus. It's actually a very complicated eukaryotic animal. It's kind of like, it's made up of the same types of cells that you and I have. And it has a very complex life cycle, as most parasites do. They have an invertebrate host, which is a mosquito, and then a vertebrate host. So it injects these guys, and they travel straight to the liver cells, and then they reproduce inside the liver cells, and then they explode as what this form now is called schizons. And they rupture, and then they go and they travel into your bloodstream, and that's where you kind of get sick. So this stage right here is asymptomatic. You don't even know you're sick at this point. It's when they get through your blood, and they enter your blood, and they reproduce inside your blood, and then they burst out of your red blood cells, and then they find more red blood cells to infect, and so on and so forth. And then at some point they split off and go into a different pathway where they form what we call gametocytes, which gametocytes are sex cells. In other words, then they differentiate between male and female at this point. So this is called kind of asexual reproduction, where they reproduce by division. And then they form these two separate male and female, which that's where the mosquito then bites again and sucks up. And then these things then form and develop inside the mosquito cycle here. And then the cycle continues, and this mosquito goes by somebody else inside. So that's how this works. Now, give you kind of a close-up of the process of these things binding to the red blood cell. I told you earlier that there's proteins that these things release that actually recognize red blood cells and bind to them, because otherwise they would be infecting all kinds of cells. They're specific to red blood cells, and they like hemoglobin. That's what they metabolize. So they bind, it sticks to it, and then that process then starts this reorientation, where then it kind of sticks itself, like I guess, head first, and then it forms what we call a junction here, and then it enters the red blood cell, and then it starts doing all kinds of funky things and reproducing. And this is a picture from my thesis where it's showing this is stained, these are red blood cells, this is a stained slide with these little things bursting out of red blood cells and trying to get ready to infect another one. So what's important to remember is malaria has been a human pathogen for thousands of years. And also, it evolved very well to infect humans, because humans have found many ways to kind of evade infection from malaria. But malaria evolves to counteract these mechanisms. It's the idea of coevolution. There are malaria sites, and the genus and species is Plasmodium falciparum is what actually is the worst of the human. There's 4 human species that are pathogenic. But there are other species that only infect, like birds, that only infect snakes, and these species can't infect us. They evolved to infect specifically. So malaria parasites also have their own lineage, evolutionary lineage. And there's probably transitional forms at a certain time. Now let me talk a little bit about sickle cell anemia. And what sickle cell anemia is, it is a genetic disorder. That's simply all it is. It's a mutation in your hemoglobin gene. That's all it is. One single point mutation in that whole gene that causes the red blood cells to kind of pancake up into this type of sickle form. And this does not hold hemoglobin and oxygen very well. You have all kinds of problems. They tend to clog up capillaries and things like that very easily. So this causes a lot of problems. Lots of symptoms, late growth, fatigue, fever, all kinds of problems with sickle cell anemia. And the way you get it, you get this trait, is, we have 2 alleles, as everybody knows, 2 copies of the genes inside your body, one copy from your mom, one copy from your dad. And what happens is if you end up, if your mom and your dad are both carriers, and you happen to end up with 2 copies of this mutation, you're going to develop sickle cell anemia. But what's more likely to happen, odd-wise, statistically, is that you're going to get 1 copy that's going to be a healthy hemoglobin gene, and 1 copy that's going to be the disease, or the mutated gene. And then, of course, there's a chance that you might get 2 copies of normal genes. And that kind of shows in the schematic here, 2 copies. So this is normal. You got 1 in 4 chances of being normal if both of your parents are carriers. 2 in 4 chances of being a carrier yourself. And then 1 in 4 chances of actually developing the disease. So, and I'll get to why this is important here in a minute. So, there I think it was about 40 or 60 years ago, I think, there was somebody studying lineages of the African people in Africa, in Kenya. And what he did is throughout his studies, he kind of started seeing that there were some patterns of sickle cell anemia in certain parts of Kenya. He noticed that in the western and the eastern part of Kenya, both of lowland and highly wet kind of areas, you had a lot of incidents of sickle cell. But if you move to the center of the country, in the highlands, there's virtually no sickle cell anemia in the populations that he was studying. He started scratching his head, like, I wonder why you have these select populations. And then what he discovered was that in the east and west coast of Kenya, it was actually where the Anopheles mosquito bred. And Anopheles mosquitoes is actually what carries malaria. So he said, you know what? I think there's got to be a correlation here. And what he discovered is that there's actually persons with sickle cell trait, okay, that have just 1 copy of the gene, seem to be more resistant to malaria than those who do not have this trait. What really is happening is that the malaria parasite exhibits a strong selective pressure. Okay, we're talking about natural selection. This is a selective pressure for the sickle cell trait. So you're thinking about, and of course this comes up at a trade-off, right, for more sickle cell disease. Here's the thing, if you have sickle cell disease, you're probably not going to make it to reproductive age, or maybe with modern medicine sort of can, but assuming we don't have modern medicine. And with malaria, you're usually not going to make it to reproductive age. But those carrying the trait are going to be immune to malaria, and they're going to live a healthy life. So evolution does not pick the best, right? It doesn't pick the best solution. It picks out of what's available. And this happens to be a good trade-off from a population standpoint. You have more people surviving malaria in areas where malaria is endemic. Why not? So these genes then are going to be selected for, because those people with malaria are going to die off, and they have the normal genes, but these guys that have this mutated gene are going to live on. So this is why we see sickle cell anemia still to this day. So how does this work? Well, several studies suggested that in one way or another, sickle hemoglobin might get in the way. So they're trying to figure out the mechanism. So they said, okay, it's going to get to the way, because malaria metabolizes hemoglobin. That's how it feeds off inside your cell. So they're thinking that a mutated hemoglobin is not going to work well with malaria. But turns out that's not the case. That's not actually what happens. As a matter of fact, malaria actually really likes the mutated hemoglobin. So, So that had some people scratching their heads. It's what we call paradoxical resistance to malaria. That's kind of an interesting aspect. This is kind of paradoxical because sickle hemoglobin makes the host actually tolerant to malaria. So you have a disease or a mutation that actually is more favorable for malaria to thrive in. So how does this work? Well, what happens is there's an enzyme whose expression is strongly induced by the sickle hemoglobin. It's called heme oxygenase, and it's part of a cascade of an anti-infection cascade, which it metabolizes hemoglobin, heme, in the blood, and produces carbon monoxide, which that carbon monoxide then stops heme from being metabolized in the bloodstream, which that then stops the malaria parasite from growing. Very complicated aspect. This work was actually figured out by Miguel Soares' laboratory in Portugal. They published a paper in Nature about this. And this is a summation slide where you have sickle cell red blood cells that produce hemoglobin heme through this cascade here. Hemoxygenase 1 is formed, which produces carbon dioxide, which stops this pathway of that infected red blood cells need to continue on and produce cerebral malaria. Now, people that have the sickle cell trait may still have parasitic load inside their body, but they don't get cerebral malaria, and that's what actually ends up killing a lot of people, a lot of kids. There's also another pathway that this does, and it stops these cytotoxic T cells from aiding malaria and spreading and binding to the capillaries in the brain. That's what cerebral malaria actually is. So that's the mechanism. On top of that, we're talking about, so now we've talked about the selective pressure. I want to talk about this idea of predictability, this predictive value that we see in evolutionary theory. As the guy 60 years ago, I forget his name, when he discovered that in Kenya, there were, on the east and west coast, that's where you find high sickle cell because that's where mosquitoes are. We have now been able to map where sickle cell is prevalent. We can actually do the study of the Wellcome Trust, which is a huge nonprofit, biomedical research nonprofit in London. They did a study with biologists and statisticians and geographers in the University of Oxford, together with the colleagues from the Camry Wellcome Trust Research Program in Kenya, have produced the first detailed global map showing the distribution of sickle cell gene, okay, and how it relates to malaria. And so these predictions, they predicted that wherever you see malaria endemic, you're going to find high frequencies of sickle cell anemia. That's the beauty of predictive value. It's not just for fun, it actually has medical value. And we see this is in humans, okay? And it's something that we couldn't have never figured these things out unless we thought and we accepted evolutionary theory and natural selection and how it works. So, is our children learning it? Well, some people still don't get it. So let me give you just another example of evolution here. You got volcanic regions in Arizona, where these volcanic regions these lava flows cut into the environment here. You can see here this black lava outcropping over a sandy desert. And unfortunately or fortunately for these guys, these pocket mice that live in the desert, they kind of got stuck when these lava flows happened because now their territory changed from sand to dark. Luckily for these guys, though, they have a mutation, a color that shows them being either black or sandy. And if you can see here, if they are over sandy terrain, they're going to live because they're going to be camouflaged. They're going to more likely live anyway. And then if you put them in the black, in the lava outcropping here, all of a sudden they're sticking out like sore thumbs. And these guys are considered to be like the Snicker bars of the desert, by the way. Everything eats these things, like everything. Like they are at the bottom of the food chain. And so, you have like these, but as you can see, so if you have a mutation of black on sandy terrains, these guys aren't going to live very well. So I want to show you a quick video on that to kind of explain it a little bit further. Hopefully. Oh, hold on.
Consider the southwestern United States. Sandy colored landscape until some lava flows happen. That lava cools. And now that black rock is going to be invaded by the sandy colored mice. Consider the southwestern United States. Sandy colored landscape until some lava flows happen. That lava cools. And now that black rock is going to be invaded by the sandy colored mice. Once every 1,000 years or so, a black mouse is going to pop up. The owls are going to be there circling overhead for eons. And if we start with a population where just one mouse is black, and it has just a 1% advantage, watch what happens over the generations. And in about 1,000 generations, 95% of the mice will be black. That's just natural selection doing the work. One mutation, one founding mutation can spread to 95% of the population in 1,000 generations. Now what happens if there's a 10% advantage of being black? Very quick. In 100 generations, 95% of the mice will be black. It turns out from measurements in the wild, there's an even greater than 10% advantage of mice being black on the black lava floors.
Okay, that was Dr. Sean Carroll. He's the Vice President for Education, for Science Education at the Howard Hughes Medical Institute, which is actually where I work at Genelia Farm. It's actually the one paying job that I have. I work for 3 nonprofits, and at least one of them is paying me. But anyway, so Dr. Sean Carroll he's an evolutionary developmental biologist, and he's actually, stay tuned because he's about to do some pretty cool stuff at HHMI as far as public education. We're spearheading, we just hired producers from National Geographic to start doing productions for us to be put out there on TV. So stuff, things like that. So anyways, so as you can see, evolution can work very quickly, and it works. And then the other, another thing I want to talk about, this is kind of one of the last examples, is this bloodless fish. In Antarctica, there's a family of fish that actually have no blood. The only vertebrate in the world without blood. How could that possibly be? Okay, they have no blood, literally. So this is about a 10-minute video that was started from the Howard Hughes Medical Institute that I'm going to show you guys because this is really cool stuff here. And it's going to underscore a couple of big things.
For life to survive, it must adapt and readapt to an ever-changing Earth. New traits and capabilities must be invented, while some traits may be lost. To see one of the most dramatic examples of how this occurs, we will follow a voyage of long ago that will take us 1,600 miles off the southern tip of Africa to one of the most remote places on the planet, Bouvet Island. A speck at the edge of the Antarctic, it's pummeled by water so wild that getting there was no sure bet, especially in 1927 when this Norwegian steamship left port.
cold, it's windy, it's rough waters, and they're far away from civilization. So it was a really long and arduous voyage from Norway to eventually the shores of Louisiana.
Norway had dispatched the expedition to claim the island as an outpost for its whalers. And after 2 months at sea, on December 1st, the crew finally rowed ashore and erected the Norwegian flag. But that's not the most important achievement of this expedition.
One aspect of their voyage was to understand all the sea life that was there. And so there was a zoologist on board, Ditlev Rustad. And Ditlev's job was to really just throw a net over the side and trawl up, see what he found. But one day, in fact, the day after Christmas, 1927, he pulls up a really unusual looking fish.
A creature that many years later would illuminate our understanding of how evolution works. A creature so unexpected, it is still barely believable today when taken from a freezer at Northeastern University by a biologist who spent much of his career trying to make sense of it.
Well, Sean, it's easy to see why the fish was originally called a crocodile fish. Notice very big protruding jaw with lots of teeth.
Incredible, but it's so translucent. It seems like I can see inside the fish.
Well, the reason you can see inside the fish is that this is a scaleless fish. And in fact, if you, well, that's the brain. You can actually see the brain through the skull. And then you can also see, projecting from the brain, the optic nerves going over to the eyes. And Sean, for comparison, we can take a look at a cod. And when I pull back the gills of the crocodile fish,
Creamy white, look at that.
and here, if we take a look at the cod,
Oh, brilliant red.
yeah, very, very good.
So that would have surprised Rustad, because all the other fish he had seen would have red gills.
Absolutely.
But that wasn't the most incredible thing about Rustad's find. That came to light when Rustad, like any good Norwegian confronted with a strange fish, filleted it, as I'm doing today under Bill Dietrich's watchful eye.
Unbelievable. Everything's white, everything's colorless. All the organs, all the soft tissues, the heart even. Every other fish he had opened in his life was full of red blood, but not this fish. And he wrote in his notebook, boldly,
Colorless blood, blue, farmless. The crocodile fish, or ice fish, as they've come to be known, had a dilute liquid almost as clear as ice water flowing through its veins.
And here's your ice fish water.
that's amazing.
Except for the ice fish, all of the vertebrates on Earth, reptiles, birds, mammals, and all other fish, like this cod, have red blood coursing through their veins.
So what's so incredible about having no red blood cells at all is that, for example, our blood, 45% of our blood by volume is red blood cells. So we have enormous numbers of red blood cells in our blood, as do most other backbone animals. And when our red blood cell count decreases a little bit, we call that anemia. And if it decreases a lot, that's life-threatening. So it's stunning that an animal could get by with no red blood cells at all.
Why would a fish abandon a way of life that's nourished its ancestors for 500 million years? It has to do with the extreme habitat in which it lives.
The more cells that are present in blood, the thicker it becomes at cold temperatures.
So this watery blood allows the fish to live in really cold temperatures. So if it had blood like this, it'd be all gummed up.
That's correct.
All right.
Blood is red because it contains hemoglobin, the protein that delivers oxygen throughout the body. Yet the ice fish have not one hemoglobin molecule. Fish absorb enough oxygen from the sea through their scaleless skin so that they can make do without hemoglobin. But But the lack of red blood cells still doesn't fully explain how any blood flows in oceans this cold, below the freezing point of fresh water and almost at the freezing point of the seawater.
And fish cannot live generally in that sort of environment because the freezing point of the blood is higher than the freezing point of seawater.
Yet fish thrive in these waters, ice fish and other species, all members of a group called the notothenioids. To show just how different Antarctic notothenioids are from other fish, husband and wife biologists Arthur DeVries and Christina Chang Dunbar in water as icy as the ocean outside their lab on Antarctica's McMurdo Sound. The temperature, minus 1.8 degrees Celsius, is so cold it's below the freezing point of fresh water and blind.
So as you can see, this fish is doing fine at this freezing temperature in the midst of ice crystals.
How these fish thrive in such cold waters baffled scientists until the 1960s, when Arthur DeVries found that Antarctic fish had invented something that protects them from freezing: antifreeze.
These fish have a certain protein, and what it does is bind to the ice crystal in the fish. And by binding to it and preventing it from growing bigger, the fish doesn't freeze.
So only one group of fish in the Southern Ocean, the notothenioids, ice fish belong to them, have antifreeze. So it's an invention unique to this group. And that's allowed those fish to invade a space that other fish couldn't live in. And these are nutrient-rich waters full of lots of food. So these fish are thriving now where other fish couldn't thrive.
Antifreeze proteins give notothenioids a clear edge where they live. But their existence poses an evolutionary mystery. When and how was antifreeze invented? The waters around Antarctica were once the temperate 10 degrees Celsius. But 34 million years ago, Drake's Passage opened as Antarctica broke away from South America. Now a continuous current circled the new continent, isolating its surrounding waters. As a result, those waters chilled to minus 1.8 degrees Celsius. So notothenioids and antifreeze evolved in the last 34 million years. But traits are encoded by genes. Where did antifreeze genes come from? DeVries, Chang, and Lingbao Chen worked out part of the mystery in the 1990s by noticing that parts of the antifreeze gene, labeled in red, strongly resembled a different gene, that one gene gave rise to the other. The process began when the ancestral gene was accidentally duplicated. While one copy remained the same, the other accumulated mutations that eventually gave it a new function: to make antifreeze proteins.
The invention of antifreeze is a crystal clear example of inventing something new from something old, borrowing sort of the code of a pre-existing gene and then altering that to create a protein that has entirely new functions. In this story we see again and again in evolution, inventing something new from the old.
Notothenioids invented antifreeze genes, but one family, the ice fish, went a step further and eliminated red blood cells and hemoglobin altogether. As for how that happened, the answer has also been preserved in DNA. And Bill Dietrich found it when he compared globin genes from ice fish and other fish. Globin genes encode the hemoglobin protein.
So what do we see here?
Well, as you go from left to right here, the normal red-blooded globin gene is shown in the sequence on the top. Down below, we have the sequence of the ice fish. And as we move from the left, you can see there are dots. These are exact matches in the sequence. However, at the arrow, we see we get virtually no match. This is essentially genetic gibberish.
So it's broken right there.
That is exactly right.
At some point in history, a mutation wrecked this gene. And since hemoglobin was no longer necessary to ice fish, the mutation wasn't weeded out.
And this is the fossilized remnants of that gene.
So if the gene has become useless, it will eventually be lost as these mutations pile up and just erase what used to be there.
That is exactly right.
So genes are born and genes die, as species find ways to survive in an ever-changing world.
This remarkable fish, the Diplophrostidae, dredged up 80 years ago, has taught us is that evolution doesn't always come up with the best solution imaginable. It just comes up with the best solution available. And that sometimes means getting rid of things that worked in our ancestors, as well as inventing new things like the antifreeze. And that record of changing habitats and changing lifestyles and changing genes is etched in the DNA record of life.
So this kind of video kind of underscores a couple of things about evolution and some of the misconceptions about, you hear a lot about people saying, well, mutations can never possibly contribute to information. A lot of times you hear genetic mutations is actually deleterious. Well, yeah, if we had genetic mutations in our hemoglobin gene, you and I, we would die. We can't survive. However, with the ice fish, that actually turned out to be a selective advantage. And over time, they lost that gene. You see it. It's there as a fossil. It's a fossilized gene living inside the DNA of these fish. And then again, so that's one aspect. But then you also saw how these fish also created a new function from proteins that already used in the system, like the antifreeze proteins that they use. Actually, the genes where these develop from actually coded for other functional proteins. And we're going to talk a little bit about that here in a minute, about how genes can kind of change. But so this is a typical example of two different types of mutations in this fish, where one function is lost, and one function is actually gained. And evolved the fish to live in such cold waters where no other fish can really live. And he can exploit now that ecosystem. So that's evolution. I want to talk a little bit about evolutionary developmental biology. Remember earlier in the talk, when we were talking about convergent evolution, I was talking about the body shapes and similar function, or how do animals really evolve similar functions and similar body shapes, even though they're not related to each other? Well, the answer is in evolutionary developmental biology and something called toolkit genes. And these are a set of genes that are very ancient. Every species of animals has these genes, and they function in the exact same way, except that, so the same toolkit genes that are responsible for an antenna of a butterfly are the same toolkit genes that are responsible for your arm. So they're relatively short genes, they're also called homeobox genes. These regulate the expression of other genes during development. So here you have 4 embryos, a fish, a reptile, a bird, and a human. At some point, you can't really tell the difference. And they all have these things called gill slits, except homeobox genes will then tell in a human that to turn these gill slits into something else, and that forms our pharyngeal area. Fruit fly. Where I work at Howard Hughes, we do a lot of fruit fly. As a matter of fact, the director of the Genelia Farm, he was the head of the fruit fly genome project. We have the largest depository of fruit fly strains anywhere in the country. They're all here at Genelia. So we do a lot of work with fruit flies. We also do a lot of work with mice. And what we see here in developmental embryos, so embryos as they're developed, they're segmented. And each one of these areas has a specific set of instructions that tell that this area could turn, for example, this blue area is going to turn into a leg and a wing. In the mouse, this blue area is going to turn into the torso area. These are all regulated by the exact same types of these toolkit genes, these homeobox genes. So genes turn on in a mouse that turn on other genes, that turn on other genes, that turn on other genes, that turn this into a mouse. Whereas here, these genes turn on other genes that turn into a fly. However, what sets off this kind of chain reaction, if you take cells from a mouse, say these red cells here, and you replace these cells here from the embryo, at this stage of development where these genes, these toolkit genes are active, what do you think is going to happen? Is this fruit fly going to turn into a mouse head? Absolutely not. The same genes, the same cells from a mouse will form a fruit fly mouth in a fly. I mean, that's pretty cool. So now imagine, when we hear that mutations don't add to complexity. Well, imagine what happens if you have mutations now in these toolkit genes that, for example, maybe allow the wings to get a little bigger. And then there's another mutation that allows this wing to get a little bigger and a little bigger. And then a little bigger after that. And we span over, maybe a million years. This thing is going to turn into a different animal, right? That's how it really works. And this is the idea of evolutionary developmental biology, is really putting the nail in the coffin for intelligent design. So, let's summarize here evolution, and we're going to step into the not-so-scientific realm of creationism. But you have a scientific theory, remember, is an analysis of a set of facts and their relation to one another. Well, all these fields of science, these sets of facts, all lead to evolutionary theory. Fossil record, genetics. I didn't talk about anything about the fossil record because everybody knows there's fossils. Everybody knows we see them, right? But that also, obviously, is another aspect of it. But genetics, we talk molecular biology, we talk, with that chemistry and physics and so on and so forth, and medicine. All these disciplines arrive at the same conclusions regarding evolution. This is independently established sets of facts. There is no conspiracy. Because that's what you hear from creationists. There's a conspiracy that the scientific community is trying to keep these people out. No, the reason why they're keeping you out is because you don't make any sense. It's not scientific, it doesn't make sense. If you made sense and you put in a paper that actually gave true and honest, unbiased evidence that really would give evidence that evolution is not true, okay, you just now got yourself a Nobel Prize. I guarantee you that. So, let's dissect this. What is creationism? Well, we go to the dictionary. It's a doctrine or theory holding that matter, the various forms of life, and the world were created by God out of nothing, and usually in a way described in Genesis. Now, there's other versions of creationism. Every culture has its creation myths. I'm going to focus strictly on the Christian creation myth that's applicable here in the U.S. Well, let's talk about a little bit the history of creation. Let's go all the way back. Okay, was Christianity historically creationist? Turns out it was not. St. Augustine, okay, he's one of the forefathers of Christianity, really. He lived in the 354-480 Common Era. Actually recognized that the Bible needs to be taken metaphorically, not allegorically. As a matter of fact, a lot of Christian apologists that are not creationists, that actually debate creationists, use this guy a lot. Say, well, no, I was St. Augustine, he's he's one of the founding fathers of Christianity. He said, no. When he was growing up, he used to be in the Manichaean religion, which is a weird amalgamation, I think, of some pagan and monotheistic ideas. But he denied the authenticity and the relevance of the Old Testament for salvation. It's only when he became a Christian and accepted the New Testament, or as the New Testament was being written, that there were problems with the Genesis, and hence was eager to help his fellow believers from getting ensnared into traps of literalism. We're talking a founding father of Christianity here, one of the founding fathers, one of the most influential, St. Augustine, is saying this. Yeah, but Catholics say this is only this. No, they're not true Christians, right? The Catholics are a different aspect. So what do we care about what these guys said, right? Oh, then come these guys. The Protestant Reformation. John Calvin and Martin Luther. These guys really shook up the creationist world, right? And those are history. So were these guys literalists and creationists? Well, Calvin and Luther stressed the need to go by Scripture alone and not by the overly rich traditional Catholic Church. That's why they were, the Reformation. It's like we need to get back from this of a man-centered Christianity to a more of a personal relationship with God Christianity, which is what is the basis of the Protestant movement today. But even they were doubtful about reading the Bible literally. Luther, justification by faith was the keystone. Okay, faith was what really made you a Christian. And yet, the Epistle of St. James seems to put greater stress on the need for good works. Well, he didn't believe in good works so much. It's faith. So he referred to it as, right strawy stuff. It's good stuff in the Bible, but we don't really need to listen to it all that much. Again, not so much a literal interpretation of the whole Bible, right? Calvin, likewise, spoke of the need for God to accommodate his writings to the unto republic. So this guy's saying, well, God had to kind of, the Holy Spirit had to kind of write this thing so that people could get it, right? So that it made sense to them. So people that aren't educated. But we can't really take it literally because talking snakes aren't really, come on, really? Talking snakes? So now we're talking about the founders of the Protestant religion are telling us that literalism and creationism should be taken word for word. So what happened? Why is it such an issue today? We're talking about the founders. This guy, Ulrich Zwingli, he was actually a radical branch at the time of the Reformation movement in Switzerland. And this guy, he put promise of God speaking directly to us through the heart, and to this day, he kind of gave rise to the Quakers. And these guys don't like a hardcore biblical centered religion. They're more about impersonal relationships. Screw the Bible. Personal relationships. So what happened? Well, what happened was the rise of creationism started during the revivals of the 18th and 19th century in America and in Britain. It's when this idea that it started becoming. This is the beginning of them becoming really political at the time. This led to a more full-blooded literalism and became a major part of the religious scenes during these revivals. In the 20th century, the Seventh-day Adventists, these guys really believe in that the earth was created in 7 days. That's what we call Seventh-day Adventists. And they really believe that these days were 24 hours. So as science started to progress in the 20th century, we started figuring things out. We said, well, that's great and all, but how does it fit with Genesis? So we got to find a way to meld this modern science with Genesis, because that's clearly the true way. So these are all aspects of what gave rise to creationism. The success of prohibition, alcohol prohibition, pushed by evangelicals. Man, they found this was like one of their early political victories. They were able to push for prohibition of alcohol. So these guys at this point, the evangelicals, they were pumped. They were out for blood. What else can we do? We're going to push our agenda to everybody now. The spread of public education and therefore evolutionary theory really gave these guys fodder to fight it. And that's where kind of in the 20th century this term fundamentalists kind of came to be. Go back to the fundamental of the Bible. And then there was this false connection, and it happens still to this day, between Darwinism and social Darwinism. Social Darwinism is a type of social engineering theory that was, I guess, used during in Nazi Germany. Or, where they use this idea of survival of the fittest, superior races and things like that. So, clearly, that's Darwinian, right? Oh, why are we going to teach this racism to our kids in public school? Well, that's not exactly what evolution and Darwinism really is. It just tended to take that form. And even to this day, my brother in college had a roommate that was super religious, and this guy had a t-shirt on that says, Darwinism is racism. Why are we going to teach that to our kids? I mean, but it's this type of mentality, this twisting of the definitions and twisting and false connections. So this all came to a head at the Scopes trials in 1925, the monkey trials, the famous Scopes trials, right? Where you had, this guy, John Thomas Scopes, a biology teacher that accepted evolution and was teaching it in his classroom against state law. It was, at the time, this was state law that it was illegal to teach evolution in schools. Well, it stirred a huge media storm. And he was found guilty, but got overturned on a technicality and an appeal. So this was considered kind of the peak of creationism. How it kind of, it really raised the public awareness of creationism and intelligent design, in creationism. But this had long-lasting effects that we still see today. So then we move into the creation science. I talked earlier that the First Amendment right prohibits, through court cases, teaching science or creationism in schools. So what happened? How did creation become science? Or how did we develop this idea of creation science? Well, 1957 Sputnik moment in Russia, we were in the middle of the Cold War. These guys were beating us to the punch on everything, going to space. So in 1958, they said, oh, no, we can't do it. We got to have a National Defense Education Act. We have to train our students and educate our children so that they can grow up and compete with Russia. And in 1963, there was this biological science curriculum study that came out from the government. And over half of the nation's high schools used these textbooks, guidelines. And evolution was part of these guidelines because, smart people realized we want to be competitive in the world. We need to educate our children properly. And then in the 1960s, as a result of all this, creation science emerges as an organized movement. And in 1970, this Institute for Creation Research, which is still active today, was formed. And the Institute for Creation Research developed creationism as a science. How do I know? You put ology behind everything and it makes it scientific, right? Clearly, that's so. Baraminology, and I'll talk a little bit about it, is Hebrew for baramin, which means created kind. So created kindology. They have things called flood geology, and creation geophysics, and creation cosmology. And of course, the latest is intelligent design. And so this is their way of convincing the religious people that creation actually can be studied scientifically. Okay. I'm going to talk about these three and focus on this. What is baraminology? There's a baraminology study group, by the way. It used to be called baraminology, but now it's called a creation biology study group. Founded in 1996. It's a group, is a fellowship of researchers and scholars who seek to develop, this from their website, a new view of biology that is consistent with the biblical record, encourage high-quality creation biology and baraminology research, and develop a community of creation biologists who share these goals. Now, baraminology, what it really is, it's the creationist version of systematics. Okay? It's the phylogenetic biology. How does it work? Well, there's a website called Objective Ministries. Unlike the racist philosophy of evolutionism, you'll see how this is actually kind of ironic that they use this term, which holds that tribes of man are diverging into different unhuman species, because clearly that's what evolution is saying, right? We're going to become an unhuman species here in the next couple hundred years. Creation science shows very well, through very nice, pretty little pictures, that a human will always be a human and a dog will always be a dog. Oh, got me sold, right? So, it explains the creation model of that diversity and the diversification of life. They have an explanation. Through the use of something called discontinuity systematics. See how they, man, I mean, this sounds really hardcore stuff. I mean, this is science. Discontinuity systematics, man, this guy knows what he's talking about. He's throwing big words at me. It shows that the pattern of life is marked first and foremost by discontinuity. Meaning that humans are in one baramin, in one created kind group, and they do not form a continuity with like the apes, because those are part of another created kind group. So there could possibly be no continuity between these two groups, right? It could possibly be related to each other. Like what? How do they know? Well, look at this. They have pictures. They have this thing called a baraminogram, where it shows you got these things, a holobaramin, and a monobaramin, and an apobaramin, and a polybaramin, and an archibaramin. I like the archibaramin. Archi stands for really old, right? It's ancient, like archaeology is the study of ancient things. Adam and Eve are in an archibaramin, separate, of course, from the caucasoids and the mongoloids. Okay, these are the human species. You got the negroids sticking out here for some reason. And then the dogs are here. Chihuahuas and poodles obviously make up one baramin, and collies and lassie. I didn't realize lassie was actually a species. I thought it was the name of it. What do I know?
Okay, and those, you didn't just make those up, right?
No. Go to the website. Go to this website here. That's where I got these from. Wow. No, go to this website. Oh, but there's more. There's more pretty graphs. More science and stuff. Biological character space. Okay? what this shows is a 3D cross-section. You know, it's three-dimensional. You know, just for added extra craziness. Of hyperdimensional continuum. Holy crap, these guys are really on something. You have apes in one group. These are potential regions, potentiality regions, where groups can be, you know, gorillas, bonobos, and chimpanzees. Here's where it's interesting. Over here, you have Negroids, Caucasoids, and Mongoloids. As if to say that gorillas, bonobos, and chimpanzees are clearly races of each other. Either that, or if you read this the other way, Negroids, Caucasoids, and Mongoloids are different species. Take your pick. What is impossible, though, is this area of discontinuity. You can't possibly have a humanity. That's not within the area of continuity. Now, you know, this sounds a little funny, but actually, you know, these creation science people are getting really crafty. They're using statistical analysis now. They're getting really actually kind of intricate and really confusing things. Where's the evidence? How's the scientific method applied here? Well, you know, if you go to the National Center for Science Education, NCSE, I recommend everybody to go visit that site. It's a great site. They actually dissect a lot of this stuff, and I took some of this from here. But it says, where's the evidence? There's no published peer-reviewed papers for, you know, these baraminology systematics. It amounts to little more than a parroting of scientific investigations into phylogenetics. And actually, their software model that they use, if you look at it objectively, the programming algorithm suggests that the method does not actually work. You know, they tweak it. Because even they admit, this guy Wood, who's a big baraminologist, will say that actually our model only works if you put miracles into account. Right? But we already established the definition of science does not involve itself with the supernatural. So anyways, this is where they really, their foundation. This is where they get it. This is where it really all goes down. All right, flood geology. I mean, just a bunch of things here. You know, they got runaway subduction. That's their geology to explain the flood. You know, all the fossils were buried within a year's time. This vapor canopy theory here, where, you know, before, in the Bible, before there was the flood, there was no rain. It was all cut up in this huge vapor cloud that actually, it filtered out UV light. You know, go figure, right? It was very clean. And then the flood happened, and boom. That's how we were able to kind of go over here. Flood geology. Continents before the flood were all amassed together in like Pangea. That's what they say. And then after the flood, they quickly, through this idea of runaway subduction, where, you know, continental drift actually happened very fast in the span of a year. So we went from Pangea to what we see the continents today, all within about a year. So let's talk about intelligent design, because this is the one that actually is the tricky one. Discovery Institute, founded in 1990. Huge non-profit think tank. These guys are serious. They're well funded. They actually have bona fide chemists, physicists, geologists, and even biologists that are published and peer-reviewed, by the way, that actually work for these guys. Of course, their work in creation science and intelligent design is never published and peer-reviewed, except their own peer review. They develop their own conferences. They develop their own people. Right? According to Intelligent Design, a scientific research program as well as a community of scientists, philosophers, and other scholars who seek evidence for design in nature, the theory of intelligent design holds that certain features of the universe and of living things are best explained by an intelligent cause, not an indirect process such as natural selection. Okay. Michael Behe, Lehigh University professor, biochemist. Smart guy, actually, very smart guy. He developed this idea of irreducible complexity, meaning that at the molecular level, biological systems can't be broken down any further. The poster child for reducible complexity is the bacterial flagellum. At the Dover trial, they talked, the creation of the intelligent design side, talked so much about the flagellum that at one point, the lawyer, they were taking a recess, and the lawyer went to the judge and said, when we come back from recess, I'm going to talk once again about the flagellum, and the judge actually was sort of saying, "Oh, goody," you know, because that's really the only thing they really had that was really. So this is a poster child. So the flagella is made up of a bunch of different proteins here. Okay, this is the long flappy thing. You know, everybody knows what flagella is. This is what bacteria use to kind of move around. There's a motor here that spins it, and they do all kinds of things. So this is a cell membrane here. This is the outer membrane, the inner membrane. You have an intermembrane domain of proteins, and the long protein here and a bunch of other protein here. These are the rotor and the motors. So it is correct that if you remove any one of these proteins, this thing is not going to work anymore. That is correct. That is a true statement. But is it really reducible? So here's the thing. You have a biomechanical or biochemical machine. It has a function. What irreducible complexity really says that the individual parts don't have a function. In other words, these were assembled purposely, and each individual piece alone is not going to work. What evolutionary theory says that you do have original components, as we've seen with like the ice fish and the antifreeze. Those proteins that were mutated actually function in things like blood clotting and things like that. They do have other functions, but they come together and then they form intermediates, which may have new functions. And then these may come together and form what we see now, for example, a flagella. So, design and function. Which one is it? Parts are useless on their own. Parts have other functions. i.e. predicts. Now, see, they're making. Remember when I was talking about predictive value earlier? They're making predictions. So the question becomes, oh, now this is falsifiable. So this is science, right? Evolution predicts that parts do have functional capabilities. You got one champion in one, and one on the other. Let's talk about this. Never mind all this jibber jabber here. Just pay attention to the colors, pretty colors. This is the flagella, okay? And this is the cell membrane here. What this is here, okay, is what we call a type 3 secretory system. And this is a system that certain bacteria use to pump proteins into other cells. The bubonic plague, Yersinia pestis, uses this mechanism to get you sick. They inject really nasty, you got this long tube. If you look at, and these are two completely different functional parts of the cell. But if you look at, if you knock off this part here, and you actually see these proteins, they have homology. In other words, the flagella evolved from proteins that had other functions in the cell. And this has been shown and proven over and over. This homology. These are all very similar domains. And they're set up. Look at how it's set up. Almost identical. So there is function. And if you cut this off, you remove this, you have this. It is function. It just functions in a different way. So what was shown at the Dover trial, and Kenneth Miller showed this, and Kenneth Miller, by the way, he's a brilliant scientist, brilliant biologist, and evolutionary biologist, and a Catholic. But we all know that Catholic's really not true Christianity. But he was able to show this to the judge, and it was, yeah, absolutely. And that's why the verdict came out for intelligent design. But here's the thing. So, yes, intelligent design is falsifiable, right? It's been falsified. So if you want to teach it in schools, I say go ahead and teach it, but show how it's falsified. Show how it's wrong. So, is there evidence? No. Is ID falsifiable? Yeah, and it's been falsified. And what can we predict from ID? Absolutely nothing. It offers us no predictive. We can make predictions, but when we test these, falsified. So I'll be all for about teaching intelligent design in schools, so long as you teach it objectively and truthfully for what it is. So in conclusion, and should creationism and evolution be given equal validity in the eyes of science? Absolutely no. What about the eyes of philosophy? That's a whole different argument. And I hate philosophy. That's an equal. How do you feel about, actually I don't really hate it, but how do you feel about teaching of creationism alongside evolution of objects? Absolutely not. Because it's not science. And it's strictly a religious entity. And should creationism be used as a basis for legislation? Absolutely not. So, three quotes I'm going to leave you with. Well, not really one. But, oh, you have evidence just for evolution. Well, you should write it down, get it peer reviewed, and collect your Nobel Prize. Teach a child to read, and he or her will be able to pass a literacy test. Really? But this is the quote I really want to leave you. There's grandeur in this view of life. With several powers having been originally breathed into a few forms or into one, And that wheels this planet has gone cycling on according to fixed laws of gravity. From so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being equal. So I just want to thank a couple of people, Sean Carroll, for he actually vetted my presentation at HHMI, and the directors of Beltway, Atheist, Ellen, Jeff, and Ray, who's the CEO. And then Jason Osborne, he works with me at HHMI, but he's the executive director of PaleoQuest.org. And go to this website. They're doing some really cool stuff. They're redefining well evolution. He's an amateur fossil hunter that is now developing methods and tools to extract fossils that are redefining not only well evolution, but also the theory. And these aren't fossils. The Smithsonian Institute tapped these guys because the director of Smith has a theory that the first Americans here in this country, in the Americas, didn't come from the bridge in Alaska, but actually came across the ocean. And they want to prove that. And these guys are in the process. So check that out. He's a great guy. So that's it for me. I really thank you. Sorry, kind of went a little long, but.
I want to thank Larry for a wonderful talk. And on behalf of Loud Atheist, I want to thank you for coming.
I had Hippocrates, I met Siddhartha the Buddha, and he walked with me. He let me go, but he never had forgotten me. I talked to Socrates, we talked philosophy, until he got to me. I told him not to bother me. Talked to some prophets, but everything they taught to me could have been gotten by a frontal lobotomy. They brought me darkness, and in this darkness it dawned on me the amazing gift Copernicus brought to me. Thus began my apostasy of prophecy, but it gave me most of all the art we see. I was there, Newton saw the fruit drop on me when science succeeded philosophy's monopoly.
Science is music. Like a child we use